Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Bayesian Outcome Adaptive Randomization Trial Designs, a Promise Not Without Perils. My name is Candace, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank IDDI, who developed the content for this presentation. The International Drug Development Institute, or IDDI, is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI has offices in Belgium, Boston, Massachusetts, Raleigh, North Carolina, and San Francisco, California. Now I'd like to introduce our speakers for today's event. Tomasz Berzykowski has over 25 years of experience in clinical trial design, with a particular focus in oncology. His research interests include clinical trial design, meta-analysis, validation of biomarkers and surrogate endpoints, statistical methods in oncology, and survival analysis. Our next speaker is Leandro Garcia Barrado, who has 8 years of experience in optimizing methods for development and validation of biomarker-based diagnostic tests and Bayesian outcome adaptive designs. His research interests include Bayesian statistics, development and validation of diagnostic tests, adaptive clinical trial designs, and crossover designs. And now, without further ado, I'd like to hand over the mic and the presentation over to our speakers, who may begin when they're ready.
Hello, this is Tomasz. Thank you for joining this webinar. We will be talking about Bayesian outcome adaptive randomization designs, and we'll take a look at several issues which are related to these designs, but also their merits. So the promise was there about a decade ago, and we can see it given here. So it has been formulated like a promise for shortening cancer drug development and better identifying responder patient populations. Now, following these initial claims, there has been a lot of enthusiasm, and you can actually see presentations like this popping up where people are saying adapt, adapt, adapt. However, after initial enthusiasm, there have been some reports related to potential issues with outcome adaptive randomization designs. For instance, this paper in Journal of Clinical Oncology by Korn and Friedlin reported that they found no benefits to outcome adaptive randomization over just classical one-to-one randomization, and they actually recommended using a fixed 2:1 randomization instead of an outcome adaptive randomization. Now, the reaction to this was like in this paper again, where it was mentioned that, we agree with the calculations, but not with the conclusions. It has been about 8 years ago, so since then we have learned a little bit more about the outcome adaptive randomization designs, and we would like to take a look at the situation as it looks now. So this is the outline of our webinar. I'll give some introduction, which will get us through the evolution of clinical trial designs towards outcome adaptive randomization designs. I'll briefly look at the previously raised issues regarding the designs. I'll introduce the example with which we will work and present the methods, and then Leandro will take over and we'll discuss a little bit more about the perils. So Our setting is an early stage, let's say phase 2 in the classical nomenclature randomized trial setting, in which we would like to check several treatments. And the goal would be to select treatments with acceptable response probability. So we assume that we have an endpoint which is binary, and traditionally in oncology, this would be like tumor response, so whether the tumor disappears or shrinks. Now, the traditional design would be like this. So we would have patients who would be randomized perhaps 1-to-1 between, let's say here, 2 treatments. We could have more. And then we'll look at the outcome. And based on this, we might want to select the treatment which seems to be promising. And if we would randomize, for instance, this trial also to a control treatment, which we could pick up the best, let's say, the most promising treatment and continue as phase 2-3 trial. Now, the idea would be to do this in a small sample size. So this selection might be problematic in terms of not really large precision of the estimation of the response probability in these groups. Now, in developments in oncology brought forward this idea of precision medicine, and we may have a situation where, for instance, a predictive biomarker may be known in another cancer type, for instance. And the idea would be, therefore, to use this knowledge to, let's say, increase the power of the trial by really targeting the patients who might benefit from a treatment. So this is the idea of the targeted clinical trial. And basically, the idea is that we would first screen the patients with respect of their assay status, and then select just the stratum in which perhaps the treatment has the biggest chance to operate, and then randomize patients within this stratum, and perhaps then we would see larger probability of response, and therefore we could actually see a clear treatment effect and perhaps select the most promising treatment more reliably. Now, the idea of fixing the randomization ratio and waiting until the end of the trial with assessing the results has been taken over by this concept of outcome adaptive randomization. So the idea is to monitor the results during the trial, and then the adaptive randomization ratio, so that it prefers promising treatment, so that we can put more patients on this treatment, with the idea that we could actually have a more precise estimation of the probability of response for this treatment, but also that we could perhaps select the treatments earlier. So in this context, we would have our patients who would be entering the trial, and initially perhaps we would randomize them 1-to-1 between our treatments, let's say in this case 2 treatments. We look at the outcome, and then let's say after this initial phase, we might decide about, for instance, switching the randomization ratio. So here we could say, okay, it's let's say odds for response are twice larger for treatment A, so our randomization will be 2-to-1, and then the next patient would get treatment A. We would observe its response. Well, maybe it's not a response, so we would downplay the randomization ratio. Perhaps then we would put another patient on A. This patient might not respond, so we would then play with the randomization ratio and so on until we would have reached perhaps the situation where eventually more patients is put on treatment A because this offers higher probability of response, and at the same time, we would enjoy the possibility of having more precise estimation of this response on a bigger sample size. Now, the idea of targeting and this outcome adaptive randomization can be combined, and this is the idea of biomarker-based outcome adaptive randomization. And in this case, the goal might be actually to identify stratum treatment combinations with acceptable probability of response. So the idea would be to actually even look at which biomarker levels might be suitable for which treatment. So in a sense, this might be a design where you could almost like check the predictive status of a biomarker as well. So here the idea would be that based on the assay, we would split patients into strata. Let's say these may be 2 strata, biomarker negative, positive say, and then we could start again with 1-to-1 randomization within each stratum. And after this initial phase, we would evaluate the outcome. And based on the outcome, we could decide about the randomization ratio. So perhaps in stratum A1, we would decide to drop treatment A because we didn't see any response there. Of course, here we show just 2 patients. This may be a little bit more, but let's say in a smaller sample size, we could, the smaller sample size might be a little bit higher, but still small. But for treatment, for stratum 2, we might go 2-to-1 for treatment A versus B. So this means that the next patient in stratum 1 would be randomized to treatment B. In stratum 2 would be randomized to the treatment A. We'll look at the responses, and then perhaps we might decide that in stratum 1 we settle with D, but in stratum 2 we might now change the randomization ratio a little bit down as compared to 2 to 1 because the response of the patient was negative, and then we could add another patient, this time to treatment A, observe the response and so on, and we could continue until the end with the adaptive randomization in stratum 2, and eventually the outcome might be that in stratum 1 we would settle for treatment B, and in stratum 2 we would settle for treatment A, in which more patients were assigned to this treatment. So that's the idea of biomarker-based outcome adaptive randomization trial, and it sounds interesting because we have this potential of putting more patients on more promising treatment. So this is also some kind of attractive point for patients who might be willing to enter these trials as well. Now, there have been several issues already raised about this idea. One is related to the ethical considerations, and this paper puts forward a couple of these issues, like the loss of equipoise, the lack of processes for adequate informed consent, and the inequalities which are inherent in the research design because the randomization ratio changes, which could lead to perception of injustice. Now, what more important for us at least are the statistical issues that, like in this paper, it was mentioned that an outcome adaptive randomization trial will require a larger sample size and therefore take longer to complete, slightly, but still, as compared to a fixed randomization ratio trial. Also, outcome adaptive randomization results, trial designs result in a slightly larger number of patients having a bad outcome than a fixed randomization trial. Again, a bit slightly, but still, this is like a result which is probably worrying because we wouldn't like to put more patients on a bad treatment. In fact, this is what has been brought up in this paper from 2015, where it was reported that there's a high probability of a sample size imbalance in the wrong direction in outcome adaptive randomization, wherein more patients are assigned to the inferior treatment arm. And this is the opposite of what you would like to. Also, these designs produce a greatly overestimated actual treatment effect difference and also surprisingly smaller power to detect the treatment difference. So there has been some reports that actually these designs do may suffer from some, let's say, features which are not that favorable if you look at them more carefully. So we would like to look at these and other issues, and we'll present some results which we have available coming from our own research. And our own research will use this example, which comes from the paper by Barry and colleagues in 2015. We'll just look at the simple setting, 2 treatments, let's say control and experimental, with 2 strata, let's say biomarker negative and positive, and each stratum would, each treatment would have its own probability of response. Let's say that we will try to decide whether the probability of response is unacceptable. In this case, let's say 25% will be unacceptably low, and the desirable will be 50%. So 25% will be also the clinically meaningful improvement. And as in the biomarker-based outcome adaptive randomization design, we will try to identify stratum treatment combination, which gives us acceptable probability of response, so hopefully something around 50%. The idea is to start initially with one-to-one randomization in both strata for, let's say, fixed sample size, and afterwards start to change the randomization ratio after every patient. Importantly, we will allow stopping for futility during the trial after each patient. So we will allow to stop treatment in a particular stratum for futility, but the efficacy will be tested only at the end of the trial. So treatment which is not stopped for futility will be continued until the end of the trial, and only at the end of the trial will be tested for efficacy. How it will be tested? Well, for this we'll use Bayesian hypothesis test. So we will be looking at the posterior probability of the treatment probability of response is, let's say, there's a low chance that it's actually acceptable. And if there's a low chance, let's say 2.5% that the treatment is maybe really working, then we will stop such a treatment for futility. On the other hand, and this can happen during the trial. On the other hand, at the end of the trial, we'll look at the posterior probability that the treatment's probability of response is higher than the unacceptable level, okay? And if this probability is large, so let's say 90%, then we'll say that the treatment shows a promise, right? And therefore we'll claim it efficacious. These are the criteria which were actually posed forward by Barry et al., but they have been used also by other researchers. Now I mentioned that there will be Bayesian testing. Well, if we operate it within a small sample size environment, we really need to squeeze out as much as possible from the small sample size. So we'll consider a hierarchical probit model, where essentially the probability of response in a particular stratum for a particular treatment is taken from a latent continuous distribution, and this distribution, let's say normal, is having a mean value specific for the stratum for the treatment. And on top of this probit model, we'll put, let's say, some distributions, some prior distributions, where we'll be saying that this mean comes from the normal distribution with mean phi t and variance sigma squared. And the mean itself, the phi t, will also come from a normal distribution with overall mean alpha and variance tau squared. Now, the first step allows borrowing information across strata for a particular treatment with sigma squared controlling the amount of this borrowing. So this will allow us to sort of like boost up information from over different strata for a particular treatment. On the other hand, this other random component will allow us to borrow information across treatments. So in a sense, in this way, we can also borrow information about which treatment about the efficacy of the treatments from each other. So this is the Bayesian framework. So essentially, using this hierarchical model and combining it with the prior opinion, we can actually get a prior opinion about the probability of response. And then we combine it with the data obtained for the particular stratum and particular treatment, and then we get our posterior, our modified opinion. And based on this posterior, we can make decisions. And as I said, we can use this posterior to decide about the futility. So in this case, if this would be our posterior, which comes from a combination of the prior opinion about our probabilities of response and the data, we could say that we are interested in this region, which is the probability that the real treatment, that the treatment probability of the response is larger than 50%, our desirable level, and we would like this probability so if this probability is low, it means that actually there's low chance for the treatment to be efficacious, and then we would stop the treatment for futility. On the other hand, for efficacy, again, after combining the prior for efficacy with the data, we could get the posterior, and we would look at this region of the posterior, which is the region for probabilities of response larger than 25%. And we would like to have this region to have high probability, like as I said, about 90%, because this would indicate that there's a high chance that this treatment is actually efficacious, that its probability of response is larger than this unacceptable level of 25%. So we have a Bayesian framework. And with the Bayesian framework, there's a lot of choices which we need to make. So if we want to implement a trial like this, we need to decide about prior opinion, what prior distributions we want to use, whether it should be conservative, naive, liberal, or would you like to use no prior opinion? One of the issues is the biomarker Si perfect, because we'll be splitting patients into the strata. So the question is, can we do this perfectly, or do we have some misclassification error? How to update the randomization ratio, and when to start adaptive randomization, after 10 patients, 20 patients, and so on. How to allow for early stopping, only for futility, as in our example, or maybe for efficacy, or both, and which stopping criteria to use. We have given an example of this criteria, but there are others which might be considered. And finally, also practical elements related to the estimation of this whole process, and criteria which are used, for instance, in the estimation of the model to, for instance, conclude the convergence. So these choices are important, and we will look at, if you think about Bayesian designs, if you look at guidance of the FDA for Bayesian statistics, actually medical device trials, but these are the guidances which you can encounter also when you discuss the Bayesian designs for drug trials. FDA will recommends that you provide the type 1 and type 2 error rates for your proposed Bayesian analysis plan. So the idea is that despite the fact that this is a Bayesian framework, we should look at the frequentist properties of these designs. So this is actually the question which we pose. How do these choices impact the operational characteristics of the trial in terms of power or in terms of type 1 error? And this is where Leandro will take over from.
Okay, thank you. So let's indeed look at some of these choices that we have to make and how these can actually affect operational characteristics of a trial. So we'll just zoom in on these three. So first of all, we'll have a look at this definition of this prior opinion. Second, we'll look at how actually the choice of our biomarker assay or the accuracy of this biomarker assay can influence operating characteristics. And finally, we'll have a look at the stopping criteria that we use. So the thresholds in this Bayesian hypothesis test. If we change that, how does this actually affect that operational characteristics of these types of trials? But before we do that, let's set the stage and start with with simulating some trials. So we will simulate trials that for which adaptive randomization will start after 25 patients. We will consider a set of maximum sample sizes, and for each of these maximum sample sizes, we will simulate 1,000 trials. In terms of underlying true response probabilities, so these are these pi parameters that we're interested in, we will define them as follows, where only the Stratum positive, so the biomarker positive patients treated with the experimental treatment will have what we would call an acceptable response probability of 50%, while in all other combinations, we will have a response probability of 25%. So when we do the simulation, we get results, and we will be focusing on actually three operational characteristics of these trials. The first one will be, like stated here, the average number of patients that are actually randomized to each of these combinations. So what you see in this graph is that for every Stratum treatment combination, so here in green you see the efficacious combination. So this is when you treat the biomarker positive patients with the experimental treatment, and you see that within this Stratum there is actually high probability or an increasing probability of being randomized to the most promising treatment. So this is actually a good thing. This is what we would expect. You see a lot more patients are randomized to this treatment than to the control treatment you see here listed at the bottom. Now, for the biomarker negative Stratum, there's something strange going on, and that seems that there is a slight, slightly more patients are being randomized to the experimental treatment. While if you would consider back what we actually had as an underlying setting, there is no difference in response probability for the Stratum. So both the control as the experimental treatment actually have the same response probability. So this is something that we need to look into. Second operational characteristic or result that we will be looking at is the probability of concluding fertility. So again, you see here that for every Stratum treatment combination as a function of the sample size of our trial, we see the probability to actually stop for fertility. And what you see again is that these red curves, so the ones which are actually the inefficacious Stratum treatment combinations, have a higher probability of stopping accrual along the way. So this is again a good thing. But what you see here, what's kind of strange, which was referring to it previous figure as well, is that there is a small probability to conclude fertility for this biomarker negative patients which receive the experimental treatment. Again, something which you would not expect because both of these treatments have the same probability of a positive response. And another thing is that in the efficacious Stratum treatment combination, you see that there's quite some large or high probability to stop for fertility as well, and this seems to increase over sample size. So this is also something that we would not want, so we have to take a look at that as well. And finally, we look at the probability of concluding efficacy, which of course for the efficacious treatment combination, this would mean that this is actually power. So this is the power of actually finding this efficacious stratum treatment combination. For the inefficacious combinations, this is type 1 error. So again, it does seem that this design is doing a good job because we have a quite large power to actually detect the efficacious treatment combination. At the same time, we have a small type 1 error, so it's around 10%, so that's also fine. But what's intriguing is that you see here that the power actually decreases with increasing sample size of our trial, which is not something that you would expect in this setting. So let's now investigate these underexposed perils. So let's first zoom in on defining this prior opinion. So as stated before, we're in this Bayesian framework, so we provide prior opinions for all our parameters of interest. So in this case, these probabilities, this response probabilities. For the fertility test, so for the test that we do to actually stop accruing patients to a particular treatment, we define our prior opinion as follows. So as this distribution, which is centered around 0.5, which spans the range between 0 and 1, but which has a preference for values close to 0.5. So you see this peak around this 50%, so that's why we say that there's prevalence for this. This means that in terms of stopping for fertility, this is what we could call a conservative prior, because if we would actually want to have a posterior opinion, which overcomes or which actually shows that there's only a small probability of this 50% or higher, then we need data which actually drags this prior to a posterior distribution, which will lead to a significant fertility test. So that's one way to look at it. Another way is also investigate how we actually do in terms of what we would call within treatment correlation. So this means let's look at the correlation between these response probabilities of different strata which are given the same treatment. So it turns out that you can calculate this, and this is based on the values that you would actually assume for our tau square and our sigma square. And for this particular prior opinion that we assumed for these results, this is actually equal to 50%. Now, in terms of stopping, this is an informative prior, which means that a particular treatment which has a probable efficacious effect will actually be less likely to be stopped irrespectively of the biomarker stratum that you're looking at. So if we want to make this concrete, in our result, so if we're looking at these two strata which are given the experimental treatment, because you have a low probability of stopping for fertility for the experimental treatment in the biomarker positive group or a biomarker positive stratum, because of this borrowing between these two strata, we also see that the probability to stop for fertility in the biomarker negative stratum is also reduced. So you see here that because these two are actually borrowing this information from each other because of this correlation that we assumed, you see that now if you would compare within the biomarker negative group, you see actually that there's a difference in probability of concluding fertility. Now we can try to fix this or just propose a different prior opinion for which the correlation would be closer to 0. And we can do this by just assuming a small value for our tau square. So if we do that, we see that we have now an intra-treatment correlation which is close to 0, so which is very small. And on the other hand, we see that we have a slightly more conservative prior opinion. So you see that the variability in this distribution is slightly reduced. So we're favoring this 50% a little bit more. So if we would now use this prior opinion, our results would change as follows, where we see that because we have reduced this intra-treatment correlation, now both treatments in the biomarker negative stratum actually have about the same probability to conclude fertility. And this is also seen now here, where we look at the average number of patients which are enrolled in each of these combinations, that there's no difference anymore. So there's no preference for the experimental treatment anymore. And another observation that we can make if we use this slightly modified prior opinion is that now our power is actually a little bit increased as well. Now, the fact that your prior opinion or your prior distribution can actually influence your results, even in the setting of adaptive randomization trials, is not a new idea. It was already mentioned in a paper by Zhao et al. In 2008, where they discussed the methodology of the battle trial. And what they did there is actually they looked at a setting where they have 5 marker groups, they have 4 treatments, and if we would zoom in on this treatment 1, we see that the response probability, so the true response probability of our marker group 5 is about 30%, while this is much smaller than in the other marker groups. So these are 80%, 60%. And next to that, they will assume 3 different priors, so 3 different prior opinions, which are characterized by 3 different values of sigma square. So you see that this first sigma square has an intra-treatment correlation of 99%. The second distribution has this intra-treatment correlation somewhere between 99 and 1, and the last one is something which is very close to 1. So if you now would inspect the estimates you got back from this exercise, it shows that with this increasing correlation, you actually get more bias. So you see when you have a prior opinion which is heavily borrowing information from the different strata within a particular treatment, so where the correlation is almost 1, you see that you have the highest bias. And this is also reflected, of course, in the type 1 error, where if you would compare it to the, let's say, the least borrowing and the most borrowing, this is, well, a factor 2 increase in type 1 error. Now, last thing that I wanted to mention is that this is even in the setting where they would not consider early stopping. So it's not really completely comparable to what we are saying here, but still this is something which is to take into account. So last thing that I want to say for this is that it does seem that people already have been talking about this, so it has been investigated, but it's not been investigated very thoroughly, and it seems to be a little bit underexposed as well. So let's move to the imperfect biomarker assay. So the results that we've seen up to now are all based on the fact that we could perfectly assess biomarker status of our patients. So this means that we have a biomarker assay that has perfect sensitivity, perfect specificity, which means that all of the biomarker negative patients will actually be identified as such, and all the biomarker positive patients will also be identified as such, which means that we have no misclassification, and that the prevalence of our assay positive patients is the same as the prevalence of our biomarker positive patients. This also means that our underlying true response probabilities will be exactly the same as our actual response probabilities, which you would see here. So basically, because we have found all our biomarker negatives and positives, what we would see in our data set is that it's exactly the same in terms of response probabilities. Now let's see what happens if we would assume an imperfect biomarker assay. So this means that now sensitivity and specificity are not equal to 1. Sensitivity now is equal to 80%, which means that about 20% of the biomarker positive patients are actually wrongly identified as biomarker negative. Specificity is 70%, so actually stating that 30% of our biomarker negatives are actually identified as biomarker positive. So this means that if we now look here at this diagonal, we have about 25% of misclassification, and we have more misclassification of the biomarker negatives going into the assay positive group than the other way around, which now means that our prevalence, so our assay prevalence, is actually larger than our biomarker prevalence. So we have a slight overrepresentation of biomarker or assay positives in our samples now. In terms of response probabilities, of course, this makes, now you see this in red, so this makes a difference, where if we would zoom in on the assay positive group, so the assay positive stratum, where the response probability of the biomarker positive group would have been 50%. Now it's actually 43%. And the reason is, of course, because of the misclassification in this assay positive group, there are actually biomarker negative patients present as well. So this, and they have, of course, only a response probability of 25% to experimental treatment. so actually dragging this overall actual response probability down to 43. The opposite, of course, happens in the biomarker negative group, where misclassification of biomarker positive patients actually increases the actual response probability to 31 instead of 25%. For the control treatment, we see no difference because, well, in this setting, both biomarker positive and biomarker negative patients have exactly the same response probability to the control treatment. So there, these actual response probabilities are not affected by misclassification. So if we now would compare results based on our perfect assay, which we already saw before, and our imperfect assay, we would see that in the average number of patients that actually get assigned to each of the treatment combinations, we see now a decrease in the number of patients which are actually assigned to the efficacious treatment arm within the assay positive group. You see the opposite happening for the assay negative group, which is now giving the experimental treatment, while due to this misclassification, they have an increased response probability. This, of course, affects the probability to conclude efficacy as well. So this means that power is actually significantly reduced if you look in terms of the difference between a perfect and imperfect assay, while for, again, the assay negative stratum, which is giving the experimental treatment, you see actually an inflation of type 1 errors. So now we have this inflation, and the reason is of course, again, due to this misclassification. The actual response probabilities that we observe in our data are not the same anymore, which are actually there in our population. So this in terms of this imperfect biomarker assay. Let's go to the final decision that we will look into, and this is this changing of the decision criteria. So if we look again at our results, there was this strange thing that we saw, that there was an increase in the probability of actually concluding fertility for our efficacious group, so for efficacious Stratum treatment combination, which actually increased over our sample size. And we see this quite peculiar drop in power also if we look across or with increasing sample size. Now, to look further into this, let's first simplify things a little bit. So let's forget about the outcome adaptive randomization. Let's forget about the two strata and just look within one stratum, so one patient group where we have controlled treatment which has a response probability of 25%. We have an experimental treatment which has a response probability of 50%. We're still interested in the same thing, so we want to find which of these two has an acceptable response rate of 50%, and we want to exclude this response rate of 25%. The reason why we do this kind of simplification is because if we go to the simple setting, we can actually calculate all our outcome metrics and not having to deal with this, the simulations anymore. So as a reminder, we had these two Bayesian hypothesis tests. So one for fertility, which was executed every time we actually saw a patient. So this means that we would stop treating patients with this particular treatment T if there was a small posterior probability, so this 2.5%, that the treatment response probability was larger than 50%. So if there's only a small probability mass to the right of 50% under our posterior distribution, we would actually stop for fertility. Now, if a particular treatment would reach the end of our trial, then we would test for efficacy. So again, we now look at the posterior for our efficacy test, and we would consider our treatment T efficacious if there's a high probability, so that's 90%, that treatment response would be larger than this 25%. So if most of our posterior probability mass lay to the right of 25%, so if this was larger than 90%, then we would actually conclude this treatment to be efficacious. Now, this would lead, as we've seen, to the following results. So what you see here, again, this cumulative probability to conclude fertility. So if we would zoom in on this red cross here, this would be the proportion of trials that would stop this efficacious treatment for fertility after 50 or fewer patients. And this probability here would be about 10%. Now, what is odd here is that you see that this increases with sample size, which, if you would think about it, does make sense. This is like, in a way, a sequential testing of fertility after each patient. So it means that after each patient, you can make, or you have a probability of making a type 2 error in this case, because we're looking at the efficacious treatment. And of course, trials which have been stopped before, we did not allow them to reopen. So it means that once a trial gets stopped, the treatment gets stopped, until the end. So this means that if you would look at this cross again, it means that there is a probability to actually stop here for fertility. But every trial that has been stopped before will not reach this point anymore. So they will be included here in this 10% as well. If you would look at the probability to conclude efficacy, so the figure here on the right, and we again zoom in on this red X, this is the proportion of trials that actually conclude efficacy of the efficacious treatment after 50 patients. So you see here that this probability is around 90%. And we see also again this drop when sample size increases. Well, if you compare this to what we saw in the picture to the left, this can be understood as the fact that, well, for a trial to actually be tested for efficacy, it actually needs to reach the end of the target sample size. So for this particular setting here, you would actually need to have included 50 patients to this treatment before you would actually conclude or do the test for efficacy. So this means that if 10% of your trials get stopped before that, these 10% of trials will definitely not be tested for efficacy. So they will be inefficacious or will test not efficacious by definition. So what happens here is that because we have this large or this high probability of actually stopping for fertility, we kind of put a maximum on the power that we can actually reach at the end of the trial. And so this is increasing this probability of concluding fertility. We see actually a drop in our power if we consider larger sample sizes. Now we can try to correct this or try to do something different by, if we look back at these results, by be a little more strict here so that we don't stop so often for fertility for our efficacious treatment. And if we would look here at the type 1 error, you see that it's about almost 20% here. We can also try to be a little more strict in concluding something as efficacious to try and reduce this type 1 error here a little bit. So we do this by changing actually the thresholds in our decision criteria. So we reduce this 2.5% to 0.5%, which makes our stopping for fertility a bit stricter, so it will be harder to actually stop for fertility. And we increase this large probability, so this 90% to 95%, where we make it harder to actually conclude efficacy at the end of the trial. If we now would compare these results again, you see that indeed we have reduced the probability to stop for fertility if we made it a little bit harder to stop for fertility. You see that this line now is much flatter, still a little bit increasing, but it's much less, so this probability is much lower than before. You also see that we have kind of lowered the probability to stop the inefficacious treatment, so the control treatment here, but it's still quite a high probability, so that still looks okay. In terms of the probability to conclude efficacy, so in terms of power and type 1 error, we looked at being, because we have actually here this flatter curve, so we have a smaller probability to stop for fertility along the way. We actually have the fact now that this is not really decreasing power anymore over larger sample sizes. And although we are a little bit more stricter in concluding efficacy, this power actually, with a bit higher than what we saw before with the previous criteria. And also the fact that we made a little bit harder to conclude efficacy now also reduced our type 1 error a little bit. So now we're actually around this 5% level. So these were the topics that we wanted to discuss. So let's go into wrap this webinar up in a small discussion and conclusion part. So as we can state that these Bayesian biomarker-driven outcome-adaptive randomization designs, they were introduced with great promises. For example, maximizing randomization to the most promising treatment, simultaneous predictive biomarker discovery, as well as treatment response estimation. They promised a high flexibility, and this especially in complex settings. But it turns out that this flexibility comes with perilous complexity itself. We have seen this in terms of these ethical considerations. We also saw that there were already some methodological issues raised as well. Even logistics are quite complex in these types of settings. Think about organizing such a trial where you have these 5 markers and 4 treatments, and you have to change or adapt the randomization ratios after every patient and do this in multiple centers. And eventually we have this long list of all these non-trivial choices that you have to make even before you can think about running the trial, for which we discussed 3 of them. And what we saw is that these choices, depending on the choices that you make, may actually lead to counterintuitive and unwanted trial operating characteristics. In terms of formulating our prior opinion, this borrowing may actually lead to an increase of patients randomized to the non-efficacious treatment. We have seen that if you will assume an assay that's not perfectly accurate to assign our biomarker status, we can actually have a significant reduction in power and at the same time a significant translation of type 1 error. And in the end, we saw that, well, actually defining these different decision criteria may cause a counterintuitive drop in power for larger sample sizes. And the reason was that we have also this increased probability of stopping efficacious treatment. What we also saw in terms of concluding remarks is that we actually zoomed in on these 3 different choices that we made, and all of these 3 in some or other way affected our power. So this means that also different combinations of these choices may actually counteract the design or can counteract in terms of these operating characteristics, which means that you will probably need massive exploratory simulation studies to investigate all of these possible settings, which may be, of course, very computer intensive if you go to more complex settings. Finally, also, well, we can discuss all of these choices, but what's still truly important is, of course, the assumptions that we can make about underlying true population characteristics. And unfortunately, this will never be known before the trial. So we always have to take those into account as well and think very hard about them. So I think in conclusion, we can say that the decision to actually apply these biomarker-based outcome-adaptive randomization design may require careful reflection before you embark on an invention like that. So this brings me to the end of this webinar, where I've listed all these references, and I would like to thank you for your attention. And I would now give the floor back in order to have a Q&A session.
Thank you very much to our presenters for that very insightful presentation. At this time, I'd like to invite our audience to continue sending their questions or comments right now using the questions window for the Q&A portion of this webinar. I've already received some questions, so I'll start with those. So here, with regards to the futility test, you put tau squared close to 0, which reduces the between strata correlation to 0. But it also means a lot of borrowing information across treatments. So is this a wise choice?
Well, maybe I'll take this. This is Tomasz. Well, in principle, what you could do is you could also play with tau squared and sigma squared in such a way that let's say your tau squared is much smaller than sigma squared, but not exactly 0, so that this correlation coefficient is still small. but yet you don't actually put the tau squared at the value very close to 0. So there is a range of possibilities which you could investigate there and try to limit this amount of correlation, like as you call it, Leandro, within the treatment correlation. So there are choices to be made, and it doesn't have to be the one which we just showed. We just showed an example which illustrates the issue itself.
Thank you for clarifying that. The next question here is, could an imperfect biomarker assay actually help then by increasing the power?
Well, that's something to think about, because of course, if you look at that, there are, well, let's say opposing powers that actually drive these effects that we see. Because on the one hand, if you would assume these difference in the sensitivity and specificity, you have of course, the difference in prevalence, which may put more patients where you actually want them. So this is then helping you. On the other hand, we've seen that these response probabilities are affected. So if there is a big difference between these response probabilities of the patients that are actually misclassified, then of course this will work against you. So there actually may be specific settings where it could actually help you. But that, I think, still remains to be seen and investigated. But it is an interesting question.
Yeah, and my point was that probably you would, well, I mean, in order to sort of like take this into account when designing a trial, you would need to know this, right? And that's a challenge, because we may never be in a situation of be able to sort of like know that this may be a situation. So that's actually not a situation. I wouldn't say that this is something that you could explore to boost up the power of your trial.
Lots to consider there. The next question here is, changing the thresholds in the decision criteria addresses the power issue, but it makes stopping for futility less likely, especially for smaller sample sizes. So are there other criteria that would be less vulnerable to this problem?
Well, there are, as we've shown as well, there are very many ways of actually doing the same thing, in which I mean that you could try, for example, considering different prior opinion distributions and affect the way that you will be stopping for fertility that way. Another way would be to elongate the period of time that you actually put patients to a one-to-one equal randomization, because this will actually only start and still stopping for fertility when you have already gathered more information. So that would also kind of safeguard you from stopping efficacious trials too fast. But again, it's because of this interaction of all these different choices that it's really hard to predict upfront what type of let's say strategies would help the best in what you're trying to achieve.
Yeah, but I think the question goes about the criteria themselves, right? The way they were formulated, right? Now there are different ways. There's an alternative to tell them, like not the posterior probability of being 90% over, let's say, being unacceptable, being at least acceptable or better than acceptable. You could formulate it differently, but I think that the issue here is that it's still within the Bayesian framework, where you try to combine, let's say, prior with a limited amount of data into the posterior. And I believe that whatever criterion there might be, there will be this issue that the randomness in the small sample size may still make your posterior distribution pretty random. And because of this, you might actually encounter a similar issue regarding the performance of the fertility criteria or efficacy criteria. I mean, somewhat expressing these issues which we have seen in our example. So I would say that this is very much linked to the Bayesian framework. And maybe, well, any criterion we would consider would essentially have, well, might actually be subject to the same problem. Yep.
Thank you for that response. The next question here is, would there be any expected benefit in considering this type of approach in a phase 3 setting?
Well, that's a challenge. I would say phase 3 is, well, obviously well, I mean, it's confirmatory phase, and there I think that the issue of selecting and looking constantly at the data and so on, it will be more challenging. So I would say that well, as we said, this is the introductory, well, early phase designs, and I would say that they should stay like this.
Thank you for that response. The next question here is, you began by stating what follows pertains to phase 2 trials with an active control, that is, comparing 2 or more active treatments. So the question is, do the same considerations pertain when one investigational treatment is being compared to placebo?
Well, I mean, I'm not sure whether if you really would like to put their placebo, then you would like to screen against placebo in a sense, right? So testing for futility for placebo would be, I think, pretty obvious, right? Although I might say that you might have treatment which have very low response probability, okay, active but very low. Perhaps this is something where you know the response probability is, let's say, 5% or something, so better than placebo, but not much better. And then in this situation, of course, you might have a problem like this again, because this issue, especially if we look at, for instance, for this imperfect biomarker issue, I mean, then you would again have this mixing of patients from different biomarker status. This would draw downwards, for instance, your comparison of your active treatments to the standard of care, and then you would be losing power and so on. So I would say that a priori I wouldn't probably include the placebo as one of the treatments in the outcome adaptive design, but certainly if you would say I'm close to that because of the low probability of response, then I guess everything what we're considering could apply there as well.
Thank you for that response. Our next question here is, you have shown that the operating characteristics of a Bayesian adaptive design trial depend crucially on the choice of priors. So doesn't that imply that these priors should be provided when reporting the results of the trial? But yet, this is not routinely done, and the audience member refers to the I-SPY 2 papers in the New England Journal of Medicine, for example.
Well, I think I would say yes. I can understand also that this may be difficult, right? Because in a trial like I-SPY, I guess the amount of, I mean, the number of priors which are used is huge. So it's, I mean, reporting them is one thing, but also the question about how to, you know, decide about what influence they might have on your operational characteristics of the trial is another thing, right? Because it's not only, as we said here, for instance, it's not only what priors you take separately, but for instance, what is the, like what was Leandro showing, correlation between the potential treatment effect estimates for, or treatment, or implied it's a correlation between the treatment effects for, like, across strata for the same treatment. And even if someone reports that, it doesn't mean that it will be clear from reporting that this would be, that we would know what it does. It's, in fact, one would have to sort of like do more and show these simulations to show how the correlation might look like. So it's a good point, and of course I would say yes. At least we should have a clarity about what priors are used, right? But I would say that this is a step in the right direction, but even more would have to be reported, like the kind of information about sensitivity checks of how these priors were influencing, you know, these different elements which we have been investigating with Leandro. I guess that's the first statement.
Yes, I totally agree.
Well, thank you very much for that response. At this time, we've run out of reached the end of the question and answer portion of this webinar. If you have any further questions, please direct them to the contact information showing on your screen. I'd like to thank everyone for participating in today's conference. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve our webinars. Now, please join us in thanking our speakers, Tomasz Borzykowski and Leandro Garcia-Barado, and feel free to share the recorded version of this webinar when it becomes available using the link in the chat box. We hope you found this conference informative. Have a great day, everyone.